Everyone. Today we are starting the subject microbiology. The first session of microbio is the general microbiology. Second one is the bacteriology. Third is the immunology. Then virology, mycology, and lastly parasitology. By this sequence, we are proceeding in our microbiology sessions. In introduction part of microbiology, we are studying about the various authors. First one is the Louis Pasteur, which is a father of our microbiology, and this is the MCQ point of view. This section is very important. Uh, Louis Pasteur proposed the fermentation theory, and Louis Pasteur also disapproved the abiogenesis theory is disapproved by the Louis Pasteur, which is very important point I have mentioned here regarding the NEET MDS as well as our BDS MCQs. Louis Pasteur introduced the sterilization technique, development of a steam sterilization, autoclave, and hot air oven. Louis Pasteur also developed the vaccines. Name is CAR. Mnemonic for vaccine is CAR. C for cholera vaccine, A for anthrax vaccine, R for rabies vaccine. Rabies vaccine, which is first vaccine was given by the Joseph Mister. Joseph Mister. And Louis Pasteur also gave the pasteurization technique. So, uh, about the Louis Pasteur, he was the father of the microbiology, introduced the fermentation theory, disapproved the abiogenesis theory, and also sterilization technique, autoclave technique, and hot air oven sterilization technique are given by the Louis Pasteur. Then second author is the Robert Koch. Robert Koch is the modern father of modern microbiology, and the father of, if examiner ask about the father of microbiology, then our answer should be Louis Pasteur. When they ask about the father of modern microbiology or bacteriology, then our answer is Robert Koch. Koch gives us a Koch postulates, which is very important regarding the SAQ point of view and also for the our MCQ point of view. In MCQ, they mention us the steps or 4 points about the Koch postulate, and give or mention the incorrect one statement, and we have to choose the correct statement about the Koch postulate. So first statement is the organism should be directly associated with the lesions of the disease, and second one is the possible to isolate the organism in the culture. Isolated organism when we inoculated in the lab, and this inoculated lab animal should lead to similar disease. It should be possible to re-isolate the organism from the diseased lab animal. There are two exceptions for the Koch postulate, which is the Mycobacterium leprae and second is the Treponema pallidum. We are going to learn In other lectures about the Palladium and the Mycobacterium leprae, here only mug up the two exceptions of the Koch postulate, that is Mycobacterium leprae and the Treponema palladium. Koch also discover the TB and the bacilli, cholera bacilli. Propose the hanging drop method, which is used for the demonstrating the motility of an organism. He also proposed the solid media concept. If examiner ask about the solid media concept proposed by, then our answer should be Robert Koch. Then third organism, which is the three organism or four organism associated with the Robert Koch. First one is the TB and the bacilli. Two exceptions of the Koch postulate is the Mycobacterium leprae and the T. palladium. Our third author is the Paul Ehrlich. Paul Ehrlich is the father of the chemotherapy, also called as a father of a chemotherapy. Father of a modern microbiology is the Robert Koch. Father of microbiology is the Louis Pasteur. And the father of chemotherapy is the Paul Ehrlich. Paul Ehrlich also proposed the acid fastening, gave the concept of magic bullets, arsenic based compounds against the syphilis. Our next author is the Anton von Leeuwenhoek, which is a father of microbiology. Father of microbiology. Father of microscopy. The father of microscopy is the Anton von Leeuwenhoek. He also coined the term little animalculus. Coined the term little animalculus. Then our next author is the Edward Jenner. Edward Jenner, which is a father of vaccination. Father of microscopy is Anton von Leeuwenhoek, and father of vaccination is the Edward Jenner. Give the smallpox vaccine. They were asked about the vaccination mostly. The smallpox vaccine is given by the Edward Jenner. Then we have to study about the microscopy of a bacteria, in which we have to cover the morphology of the bacteria, capsule, flagella, and other ingredients. Firstly, there is a morphology of a bacteria. Morpho of bacteria. There are two types of bacteria which we have to study. First one is the cocci and second is the bacilli. Cocci. Cocci is the oval in shape and also spherical in shape. Shape of the cocci is oval and the spherical. Bacilli are the rod shaped bacilli. In cocci, they are the following types. Types of cocci is the diplococci. Diplococci. Second one is the streptococci. Then staphylococci. Then tet- staphylococci, tetrad, and sarcina. Diplococci. Di means two. So these are in the group of two. Streptococci. Streptococci are in the chain form. Then staphylococci. Staphylococci the clusters. They are in a cluster form. Staphylococci. Tetrad. Tetra means four. So they are in the group of four. Tetrad. And Sarcina. They are in the group of eight. Four above, four below. So these are the types of cocci: diplococci, streptococci, staphylococci, four tetrads, and sarcina. Then the bacillus. Bacillus, they are further divided into cocci, coco, bacilli. In this bacilli, there is a length is equal to the width of the bacilli. Example is the Brucella bacilli. Brucella bacilli. Length equals to width, that is square in shape. Second is the streptobacilli, where the bacilli is present in the chain form bacilli. Then Chinese letter or cuneiform type of pattern is present in the angle of each. Chinese letter type of pattern. Chinese letter type of pattern. Then Kwama shaped. Kwama shaped bacilli are there. Uh, the example of a comma shaped bacilli is the Vibrio bacilli. So these are the two morphology of the bacteria, cocci one and the bacilli. We have to move further. We have to study about the capsules of the bacteria, which is again an important one. This capsule increases the virulence of the organism and inhibit increases the virulence of the organism. That's why inhibiting the phagocytosis process. These capsules are protective against the antibacterials. Examples of capsule present in the streptococci: pneumonia, then H. influenzae, then Klebsiella, and Bacillus anthrax, which increases the risk of their infection in post-splenectomy and sickle cell anemia patient. This is very important MCQ, that the capsule presenting bacteria is which is the capsule presenting bacteria. There is a streptococcus pneumoniae, H. influenzae, Klebsiella, and anthrax, which increases the risk of the infection uh, in post splenectomy and the sickle cell anemia patients. Then demonstration of capsule. There is a negative staining by the indica ink. Serological method for the demonstration of capsule is M. Faden reaction by the anthrax bacillus by the Bacillus anthracis. There is a quilling reaction. Quilling reaction is for the pneumonia. Streptococcal pneumonia. Next entity about the microscopy of bacteria is the flagella. Flagella. Flagella is the uh, for the motility of that organism, and on the basis of the arrangement, the flagella is divided into the four types on the basis of their arrangement. First type is the monotrichous flagella. Monotrichous flagella. Second is the amphitrichous flagella. Amphitrichous flagella. Third is the lophotrichous flagella. And last one is the Peritrichus flagella. In their name itself shows about their characteristic of the motility. Mono means single, which is a single polar flagella. Monotrichus is the single polar. Amphi means both. At the both end, there is a presence of a flagella. Suppose this is the bacteria. In monotrichus bacteria, there is a at the one end of flagella is present. And the example of this is the Vibrio cholerae is the perfect example of the monotrichus bacteria. Then Amphitrichus flagella is amphi means flagella at the both end. Here also and here also flagella present. Aphecalis is the example of a amphitrichus one. Then Lophotrichus. Lopho means tuft. Lopho means tuft. Tuft of the flagella. This is the bacteria and there is a tuft of the flagella. And example of the Lophotrichus is this spirilla. Then Peri means around. Around the bacteria there is a flagella. Then example of the Peritrichus is the Salmonella typhi. Salmonella typhi. So there is a four types of flagella: monotrichus, amphitrichus, trophotrichus, peritrichus. Mono means single. Example of the monotrichus single polar flagella is the Vibrio cholerae. Then amphitrichus is the Aphecalis, Lophotrichus is the Spirilla, and the Peritrichus is the Salmonella typhi. This is a very important characteristic of the bacteria, and which is asked in the tabulate form. They were giving the four options about the types and four examples of the bacteria name, and they have to match to ask about the matching of the uh, similar type of a motility to that bacteria, and we have to choose the ABCD one. So remember this is very important entity. I hope you all are enjoying our session. In another lecture we have to study about the sporulation and the growth of the bacteria. Stay tuned. Bye bye, happy studying.